Hi everyone, welcome back to Mito's Bio. So today's lesson is going to be about the cell cycle, and this includes everything like mitosis, binary fission, kind of where cancer cells come from, includes all of that content, and this is really important to know in general for biology. There's a lot of things that kind of depend on the cell cycle. And you're going to see a lot of problems about it, lots of questions in competitions like Science Bowl, different Science Olympiad events, and USABO. So I would recommend knowing this one really well. So let's get started. First, we're going to talk about cell division. So here are kind of the key roles that cell division is going to play in different organisms. So first, for prokaryotes and unicellular eukaryotes, cell division is going to help with reproduction. And hopefully this makes sense because prokaryotes and unicellular eukaryotes are literally one cell big. So, if they divide, then they're reproducing. They just made a copy of themselves. So, that's one of the key functions. And it also helps in the formation of a multicellular eukaryote from an egg. So this is how we came to be. This is how, other animals and, plants and whatnot came to be, right? So it grows us from one cell to as big as we are. And also it continually renews and repairs different areas in fully grown multicellular eukaryotes. For example, in our bodies, the bone marrow is continually making new blood cells. You know, that's an important place. And you can just see in these pictures kind of all of these functions. You see, reproduction for this amoeba. You see growth for this sand dollar embryo, and in tissue renewal in the blood marrow, or the bone marrow. All right, so now let's talk a little bit about genetic material in the cell. So it can get pretty confusing because there's a bunch of words people like to use here, and more or less they do mean similar things, but you're using them in different contexts. So let's try to figure that out. So the genome is the DNA and genetic information in a cell, and this has to be replicated before division. So you may have heard the word genome in like the Human Genome Project, which was like a 20 some odd year long project that was meant to sequence the entire human genome. So yeah, and then there's a lot of it, right? There's 2 meters of DNA in a human cell if you just kind of stretched it taut. And the next term is chromosome, and this is kind of what you would call DNA when it's packaged. So each chromosome has about a few hundred to a few thousand genes, and these you can see under a microscope. And really, it's just a more condensed form of chromatin, which is DNA and the proteins it's wrapped around. So, DNA is wrapped around these proteins called histones, and then you've got 8 histones, and that makes a nucleosome. And really, it looks like DNA is kind of wrapped around these little beads, and that just helps provide some, organization to the structure. And then when your cell is undergoing division, it's going to condense its chromatin into a chromosome. And we'll talk a little bit more about that later. And then something else to note is that human somatic cells, which means body cells, have 46 chromosomes, whereas gametes, which is, egg or sperm, has 23 chromosomes, and this varies by species. So, fruit flies have 4 pairs of chromosomes, right? We have 23 pairs. And in fact, that's why if you've ever heard of the company 23andMe, who makes like genetic testing kits so you can see, your ancestry and different other things from your DNA, that's why they're called 23andMe. So yeah, here is just a cell with genetic information. I believe this might be chromosomes because they're pretty distinct, but also I can't really see too much shape here, so I'm not sure. All right, so now let's look at chromosomes during cell division. So, like we said, chromosomes, they condense from chromatin. And this happens after the DNA in a cell is replicated to prepare for cell division. And this is something important to note. When you're dividing a cell and you expect equal cells at the end that match like the parent cell, then we're going to have to replicate the DNA, right? So each daughter cell is what we call the two resultant cells. Each daughter cell has to have the same amount of DNA. So when we have our duplicated chromosomes, here is what they look like. So there are two sister chromatids. Each one of these is a sister chromatid. So you got two of them, and then they are attached with these proteins called cohesins. And we call this sister chromatid cohesion. And then this area right here where it's kind of cinched in, it's like kind of like a little waist. This is where we call the centromere, and that's the region. It's the region where chromatids are attached closest, and it's got a lot of repeated sequences, around this part, so that's kind of like an identifying factor. But if you see a picture, it's just that part where it looks like they almost kind of cross over. And then below and above the centromere, we have these arms. So we would call this an arm. This is also called an arm. So yeah. And then during cell division, our sister chromatids are going to separate. And some important terminology is just mitosis is the division of genetic material, so separating these chromosomes. Whereas cytokinesis, which we'll talk about later, is the division of cytoplasm in a cell. So here's kind of like the big overview of what happens during cell division. You've got your, this is what your cells normally like. It's going to duplicate its DNA to make those two sister chromatids stuck together. And then when you divide, these two get pulled apart into separate cells. And then, these resulting cells are the same as that original cell. And this is only showing what happens for one chromosome. Obviously, for humans, we've got far more than that, so this is happening for all of our chromosomes. Awesome. So now let's talk about the cell cycle, and we're going to get in depth in kind of what happens before and during and after division. So here are the phases of the cell cycle. We have interphase, which makes up about 90% of our cycle. So it's this teal arrow right here. And then there's three kind of big things that happen there. There's the G1 phase, which is mostly when your cell is growing. It's fresh from, a new division, and, it's making RNA, it's making proteins, it's getting bigger. And then we have our S phase, where the most important thing is our DNA gets duplicated. And then we have the G2 phase, where our cell continues to grow and it gets ready to divide. So the G's here stand for gap, and that comes from in the past when scientists would look at these cells and they're like, hey, nothing's going on, so it's like they're taking a gap from dividing. So that's where, the G comes from. But really, now we know that they're spending a lot of energy on growing and preparing to divide during those quote-unquote gap phases. And then something else to note is that there's a G0 phase, and that's where the cell doesn't divide at all. And it's not like you have a G0 phase for all cells. It kind of depends on where you are. So some cells get pulled into a G0 phase, after the G1 phase, such as secretory cells in your pancreas, because they don't need to be dividing, and they just stay in that phase and just kind of do their job. So an example would be like neurons in your brain, because, you have already really well established connections in your brain, and that's, what kind of forms your thought processes and your memories, and you don't want to be switching those out by, having your cells divide and replace each other. So yeah. And then finally, the shortest kind of 10% is the M phase, which stands for mitotic, and this is where mitosis or cell division occurs. And then, that's ended by cytokinesis. So yeah. All right, so now let's talk about mitosis in depth. So this is a very big chart. I would recommend knowing all of this pretty well, just because it shows up a lot, and it's going to be a great foundation for you. So we're starting off at G2 of interphase, and we're already starting to get ready. And our nucleus still has its nuclear envelope intact, and it still has its nucleoli. And we've got centrosomes, which are kind of these regions where we're organizing our microtubules. They've got a pair of centrioles in each, and our centrosome has already duplicated, so we've got two. And then, our chromosomes have been duplicated since the S phase was before the G2 phase. And yeah, and then keep in mind this is an animal cell. Like in a plant cell, we would not have centrosomes, right? But now we're moving on to prophase. So in prophase, we're just kind of, nearing division again. So we're going to be condensing our chromosomes. There's going to be no nucleoli. And then the mitotic spindle, which is the network of microtubules that's stretching between these centrosomes and is ultimately going to push the cell kind of to get longer and pull the chromosomes apart. That's starting to grow, and we get this thing called an aster here, which is basically just this kind of star-like array of microtubules, those short ones right there. And then, yeah, so already we get these centrosomes kind of moving apart. So again, not much is happening. I would say the big takeaway is just chromosomes condense here. And then we have prometaphase, which some people don't really recognize, but it happens between prophase and metaphase. And a lot of the things that happen during prometaphase, some people categorize as either prophase or metaphase. But basically, nuclear envelope, it disappears, and then we get our microtubules attaching to our chromosomes. And they're attaching at this point called the kinetochore, which is like a complex of proteins. And what's going to happen is we're going to get one microtubule attached on this side and another microtubule attached on this side. And then we have our non-kinetochore microtubules, whose job is to basically elongate the cell, and they're kind of attaching as well. So now we've reached metaphase, and at this point, all of our chromosomes are going to be attached and lined up at the middle at what we call the metaphase plate. And this is not a real plate, it's just like an imaginary plane, kind of like, think of it as like an imaginary table that all of these are lying on, because they're all on one straight line. And then at this point, things should be lined up really nicely and attached. And then we move on to anaphase, which lasts very short, right? It's usually only a few minutes. And then we're pulling apart these chromosomes. So we're pulling apart the sister chromatids, so one over here, one over here. And then keep in mind that when your sister chromatids are separated, we call those chromosomes again. Might be a little confusing, but yeah. And then basically what lets that happen is you've got these cohesins, attaching the sister chromatids together, right? But those cohesins are cleaved, and then we're allowed to just start pulling them apart, and our cell is getting longer, as that happens, and eventually we reach telophase or telophase, where we're kind of restoring order again. Our nucleolus and our nuclear membrane is reforming again, and then we're starting to pinch in right here at what we call a cleavage furrow, and then we're leading our way into cytokinesis, which usually happens in telophase, but is honestly a separate process than mitosis. And that's basically when we just kind of pinch off these two cells. So yeah, that was pretty involved, pretty long, but it is definitely worth it to know all of these. I remember them like PMAT, so prophase, metaphase, anaphase, telophase. I just kind of remember cytokinesis happens at the last one, and I remember prometaphase happens in between prophase and metaphase. So yeah. Awesome. Now we're going to talk about the mitotic spindle, which is that network of microtubules that is very important. So basically the mitotic spindle formally is the microtubules and the associated proteins. And once we're going to go into mitosis, a lot of the microtubule structures in this cell are actually going to disassemble, so we can use those tubulins to form our mitotic spindle. And, they're growing from the centrosome, and then we've got our asters here, which are these short microtubules, look like a star. And then, kinetochores, we have them right here. And then the microtubules that attach to the kinetochores are called kinetochore microtubules, whereas these other ones that are just kind of pushing the cell to be longer are non-kinetochore microtubules, and they overlap. And the way they push the cell apart actually is very interesting, because in that area of overlap, you've got motor proteins that are kind of walking the two microtubules apart from each other, and that's kind of pushing at these ends. And while they're doing that, these microtubules are getting longer as well. They're adding more tubulins to them, and that essentially just makes sure that they're always overlapping, so there's always more expansion to be done. And yeah, again, we've got this metaphase plate that all of these chromosomes are on. And we've got separase to cleave the cohesins in between the sister chromatids during anaphase. And then, yeah, finally, you know how we said that during anaphase we're pulling apart these sister chromatids? Well, there's kind of like two theories people had as to how they're doing that. So one is like the Pac-Man theory, which is basically like saying that the microtubules are getting shorter at this point right here, and it's just these chromosomes are kind of getting dragged in like that. And then the real mechanism is like, if you've ever been fishing, like the reel there, you're reeling in your line. So that mechanism states that you're kind of reeling in the chromosome via this microtubule, like right here. And after some experiments, it turns out it depends on the organism, and sometimes it can be both. So yeah, kind of point of contention there, but it really just depends. All right, now let's talk about cytokinesis. So in animals, we have cleavage, and we form what's called a cleavage furrow with actin microfilaments kind of in a circle like this. And we've got myosin inside the cell, and it's going to kind of like, it's almost like a zip tie. You're like pulling it tighter and tighter and tighter like a drawstring until eventually, you're going to cleave the cell into two. And then in plants, we have a cell plate because our plants have cell walls. So essentially, you're going to have vesicles from the Golgi complex come in, and they're going to fuse together. And yeah, those vesicles have like the components of a cell plate, like, cellulose and stuff. And then that cell plate is going to form, and it's going to grow into a new cell wall. So yeah. Now we'll talk about binary fission, which is a type of asexual reproduction. So here we've got a bacterial cell, and we're starting to replicate our DNA starting at this origin of replication. So if you watch like the DNA replication video, we've formed this bubble here, and then we have a replication fork going both ways. And essentially, you're just replicating until this DNA becomes, bigger and bigger, and then, that is stretching out the cell. And there's also specific proteins that are stretching out the cell as well. And pretty much you are just going to end up with a big cell, twice the size with twice the amount of DNA, and then that cell is going to pinch in half. That's what happens during binary fission. And this is what unicellular organisms are going to use to reproduce. And here are some other types of nuclear division. So in dinoflagellates, what happens is basically you've got like your nuclear envelope stays intact, and you're going to have microtubules going through the envelope, and that's stretching out the cell, and then the chromosomes are going to stay within the nuclear membrane, and it's just going to stretch until it becomes two cells. And then in diatoms and some yeasts, you're literally just, it's kind of like you're undergoing mitosis within the intact nuclear envelope. And, you've got your kinetochore microtubules and your non-kinetochore microtubules that are elongating this whole thing. And yeah, so those are some kind of nice exceptions, I suppose, to know. And yeah, so now we'll talk about controlling the cell cycle because it's pretty powerful, to be able to, divide, right, for a cell to. And it's necessary to kind of keep that under control to make sure you're not trying to form too many new cells or not forming enough, stuff like that. So here is the cell cycle control system. So the way this was kind of found out was through a series of experiments, and essentially what the scientists did was they fused an S phase and G1 phase cell, and they found out that the G1 cell becomes an S phase cell. They fused an M phase and a G1 phase cell, and then the G1 phase cell enters mitosis. So what this told them is that the cell cycle control is done through molecules, and when you fuse those cells, all like those mystery molecules from the M phase and the S phase cells enter the G1 cell and induce those phases in that cell. So now we know that there's regulation of the cell cycle by checkpoints in G1, G2, and the M phases, and they're controlled by fluctuations in regulatory molecules, mainly protein kinases and cyclins. And there are multiple types of cyclins and protein kinases, but what we really need to know are, kind of this general system. So a cyclin is a protein, and cyclin-dependent kinases are kinases, so another protein, and we call them CDKs. So together they make this thing called MPF, or maturation promoting factor, which starts the M phase. And MPF, the way it starts the M phase is that it phosphorylates proteins, and it also acts on other kinases to essentially do things, right? That start the M phase, and we're going to see this kind of like cyclical fluctuation in their amounts, and it really just peaks pretty much during anaphase, during the M phase, and then at that point MPF is going to destroy its own cyclins. So this is actually really nice because it makes sure that you're like not promoting too much mitosis by pretty much destroying the cyclins so they can accumulate again for the next cycle. So yeah, that's like really important to know because this is the major way that our cell cycle is controlled. So we've got some checkpoints throughout the cell cycle to make sure we're not going through too fast or if we're not ready, So the G1 checkpoint is the most important, and pretty much if your cell makes it through this checkpoint, it's probably going to make through the rest of, cell division. But at this point, your cell may also enter the G0 phase, which, this might happen if just depending on what type of cell it is, or if your cell has like irreparable DNA damage, your cell can be like, okay, we do not want this cell dividing, go enter G0. And then there's also the S phase checkpoint, which is after you just replicated the DNA. The point there is to check for DNA damage to see if there's anything that needs to be repaired. And then there's the M checkpoint where you're checking for the attachment of kinetochores to spindle microtubules before anaphase, and this is really important. And even if you had like one microtubule that wasn't attached properly, you can't go through into anaphase. And then finally, these checkpoints are influenced by external factors, and for example, that could be growth factor, which promotes cells to divide. For example, platelet-derived growth factor binds to receptor tyrosine kinases on fibroblasts, which are a type of like cell and connective tissue. And the point of that is to pass the G1 checkpoint. So you can see that like if any part of that system is faulty, you might promote way too much growth or way too little. So now let's talk about cancer cells. So, cancer cells do not exhibit density dependent inhibition or anchorage dependence. So, let's talk about what each of those are. Density dependent inhibition basically means, in a normal system of cells, if you've got too many cells in an area, they're not going to divide because they realize, oh, hey, there's, way too large of a density of cells here. So, we're not going to divide, right? And that works out really great, except for cancer cells, they don't have that sense. So they'll just keep on dividing even if there are way too many cells. And anchorage dependence is basically normal cells, they have to be anchored onto something to continue growing. But cancer cells do not, and that's really dangerous because it allows them to move sometimes. And cancer cells also don't need any growth factors to grow. They can either produce their own signals or they can straight up just get rid of that like kind of need to have some sort of signal to grow. And in general, cancer cells divide indefinitely in culture. So there is this line of culture cells called HeLa cells, and they came from a patient named Henrietta Lacks, and actually without her consent, but they are used a lot in biology now. There's a pretty interesting book about it called The Immortal Life of Henrietta Lacks. And yeah, so the term transformation means when cells in culture acquire the ability to divide indefinitely, like cancer cells. And then there's this enzyme called telomerase that keeps on replacing telomeres in the chromosomes of cancer cells that keeps on letting them divide. So telomeres are these like little caps on the end of your chromosomes, and essentially every time you duplicate your DNA, every time you make a copy of your chromosomes, you're losing a little bit of DNA on the ends. So those telomeres are kind of there to cushion it, so you're not losing too much DNA that's useful. And essentially, normal cells, I think after they divide like 50 times or so, they just stop dividing, and we call this the Hayflick limit. And that's kind of like, a safety mechanism. But cancer cells, they have telomerase that just keeps on generating those telomeres, so they can just keep on dividing indefinitely. And cancer cells, they evade triggers for apoptosis. So normally, the abnormalities that would cause a cell to become cancerous, like mutations in DNA would be caught, and then the cell would undergo apoptosis. But those cancer cells can evade that, and that is where you get a lot of cancer cell growth. And then two types of tumors. A benign tumor is when abnormal cells don't move. Like those cancer cells stay in one place. But a malignant tumor is when those abnormal cells can spread to new tissues. And in this case, we sometimes call those transformed cells. And you see excessive proliferation, unusual chromosome numbers, changes on cell surface. So if you actually look at these cancer cells under a microscope, you can tell the difference because the nuclei usually look strange compared to a normal cell's nuclei. And angiogenesis, which is basically when they make blood vessels grow towards them so they can get more resources to grow. And then metastasis, which is when cancer cells move. And then cancer cells are sometimes treated with high energy radiation or chemotherapy, and these are usually aimed at making these cells stop growing. So, for example, Taxol, which comes from the Pacific yew tree, prevents microtubule depolymerization. So, this essentially freezes the cell in mitosis because you can't manipulate those microtubules to pull apart those sister chromatids, and you can't go through with cell division. Right, that is the end of the lesson. Thank y'all guys so much for watching. I hope this helped. If it did, please leave a comment or a like, and please subscribe for more content. Thank you.